Sometimes when we want to assess the offspring of organisms, we don't want to just look at a single trait. Sometimes we're looking at multiple traits in conjunction. Right? And so when we are looking at more than one trait as a single phenotype, we need to use a bigger Punnett square. Right? By combining the different traits into a single phenotype, we're changing the probability outcomes. And so, for example, we might be looking for babies that are going to be tall and purple. Right? That's a single phenotype, tall and purple. Right? It's coming from two different traits, the trait for height and the trait for color, but we're combining them and only looking for babies that are tall and purple. Right? So we don't care about babies that are short and purple. We don't care about babies that are tall and red. We're looking only for babies that are tall and purple. And our probability storyline changes when we add that stipulation, so we need a different type of Punnett square. Yeah. And so when we are looking at these multiple trait Punnett squares, right, you can see they're quite a bit bigger. And we have to still contemplate or take into account the fact that we're talking about making gametes. Right? And so the axes on top still represent gametes. And so when this male at the top makes some sperm, it's going to take this set of genes and half of it will end up in one gamete. The other half is going to end up in a different gamete. Right? Likewise with these genes. Okay. So each gamete at the top needs to have one of every letter. Right? That's how gametes work. They have one copy of each chromosome. Right? Therefore, it needs one copy of each letter. So our gametes need one copy of each letter. When our gametes fertilize, right? So when the sperm meets this egg and makes this zygote, that zygote is now a full organism. It needs two copies of each letter. Okay? So the functionality of the Punnett square is the same. It's just now we need to incorporate two traits instead of one. Okay? So it needs to be bigger. Right? So when we're looking at this, right, again, we're still in the context of looking for babies that are tall and purple. Okay? The rules I've set up here is that the dominant trait for A is tall and the recessive trait is short. Okay? So the allele for tall is dominant over the allele for short. For B trait, right, red is the dominant allele over purple. So if I'm looking for tall and purple, right, there's only a few genotypes that will make tall and purple. Okay? So for tall, I need one big A. Okay? So it can be homozygous for that trait, or it can be heterozygous. Both of these will present as tall. Okay? And for the second trait, for color, right, since purple is recessive, it needs to be homozygous in order to look purple. So only homozygous will work for this second trait. So I'm looking for either combination here, homozygous, homozygous, or heterozygous, homozygous. Any babies with these two genotypes will have the phenotype of purple and tall that I'm looking for, or tall and purple, whichever way, right? Same thing. So looking at how to mechanically do this, right, if we zoom in on this beautiful model that I've made for you, okay, this is again the male, this is the female. Okay. So this spermie right here is made by taking the underlined letters in white. Right? And so this version ended up here, okay, and this version ended up here. That would leave my small one to end up here, right, and my other one ended up here in this case, right? So, but, right. So this is which A and B this spermie got. Okay? This spermie got this A and this B. Okay? Underlined in red, underlined in red. This spermie got this A and this B. Okay? And that leaves this spermie with this A and this B. Those are all the possible gamete combinations of A and B that this individual could make. Okay? So we've made the gametes here at the top. Over here with the female, we're making some eggies. Right? And so the first one's the white. This A with this B makes that. Okay? Second one's the red, so it's this A with this B makes this egg. Third one's the blue, so it's this A with this B makes that egg. The last one's the gold, this A with this B makes that eggy. And these are all the possible eggs that this organism could have made, considering A and B. Okay. And then just like in the smaller Punnett square, if this sperm meets this egg, it makes this baby. Right? If this sperm meets this egg, it makes this baby, so forth and so on. And so these are all the possible babies that these two organisms could make. Okay? And so going through, I'm looking for this genotype. Right? And do I have any of that genotype? Let's see. No, I'm not going to have any of that genotype. I know I won't because the female can only contribute a small a. So I'll never have a homozygous big A for this organism, right? Can't happen. So none. 0 of 16 on this. Okay. I am going to have some heterozygous homozygous pairings, right? And so I'm looking for big A, little b, little b. And so this is not that. So that whole column is not that. This is that, right? So this is that. Heterozygous, homozygous, heterozygous, homozygous, heterozygous, homozygous, heterozygous, homozygous. Right? And so all four of these are this genotype. Okay? So it's going to be 4 of 16 because none of these are that and none of these are that. So it's these four right here. And now you'll notice that if you go down these columns, right, they're all the same. And that's because the female has all the same gametes over here on this side. Right? And so I have 4 of 16, which reduces to 1 of 4. Right? So 1 out of 4 of the babies from this organism should be tall and purple, or purple and tall, same thing. And so if I had 400 babies, well, almost fell over. If I had 400 babies, I'd expect 100 of them to be tall and purple. Right? It might be 98, it might be 104. It's all the same. That's the same as 100. Cool? So big Punnett squares when we're looking at more than one trait as a single phenotype. Okay? So tall and purple, that's one phenotype. Bye.